And welcome to our webinar session today. Our webinar session is titled Charge Variant Analysis for Intact Protein Characterization in a Biopharmaceutical Development Lab, Lessons Learned and the Road Ahead. Today's presentation will be presented by Dan Bach Kristensen, Principal Scientist at Symphogen. My name is Jessica Corsino, and I will be your host for today. The zip chip is a microchip CE-based front-end separation device for mass spectrometry. It can be applied to a variety of areas, which is listed here in the application section, but today we're going to focus on the charge variant analysis application. Before we turn it over to Dan and jump into this presentation, we do have a couple of poll questions. All right, I see some responses coming in. Thank you. 73% yes. Okay, great. And then we have one more poll question here. Excellent. We see 70 or 80% ICIEF. And now to our session, introducing our speaker, Dan Bach Kristensen. Dan is a principal scientist at Symphogen, a part of the Servier group. Symphogen specializes in the development of antibodies, antibody formats, and antibody mixtures for the treatment of cancer and neuroinflammatory diseases. For the last 19 years, Dan has been working with an analytical development in the biopharmaceutical industry on projects ranging from early discovery to product registration. Clinical indications include bleeding disorders, neutropenia, autoimmune diseases, and oncology. Dan holds a PhD in biology and a Bachelor of Science degree in chemistry. Today we'll focus on the work that they're doing in their lab on charge variant analysis for intact protein characterization.
Thank you for attending my talk here in the middle of the summer vacation. I appreciate it. And today I'll be talking about how we do charge variant analysis in our lab with a focus on the zip chip technology. So before I dig into that, I just want to go a little into the background and what's motivating us and what challenges we have and what kind of tools we are using to support the biopharmaceutical development at Symphogen. Of course, this audience knows very well that when you're working with large biomolecules, large proteins such as antibodies, you have a really large and complicated molecule, and you need to understand all the product variants of your product that affect safety and efficacy of your product. Now, when you consider a normal antibody, which is around 1,300 amino acids, and you combine that with the large number of product variants and modifications these molecules can undergo, you get a combinatorial explosion of possible product variants, and you really need to consider a biopharmaceutical product as a mixture of many variants, and of course these change over time. When you leave it on the shelf at the recommended storage, what happens to the product, and these are important things to address because they will ultimately determine, for instance, the shelf life of your product. Now, we really like mass spectrometry because all these modifications have a mass signature, and today modern mass spectrometry is really capable of digging deep into those product variants, both using intact workflows or subunit workflows or peptide mapping, kind of bottom-up approaches. And also, you have an added level of complexity with the new molecular formats that are being introduced or have been introduced for a while now. You have your classical IgG molecule, which is complicated enough. Then you can bispecifics with different chains. You can have antibody-drug conjugates where you have these warheads, chemical warheads attached to a molecule, and they combine, say, for instance, a cytotoxic effect of the chemical warhead with the specificity of an antibody. But you can also combine antibodies with non-antibody parts and working with fusions. These are just some of the examples, and we are working with all these molecular formats in our lab. a view of our hardware platform, hardware and software. Now, I won't go into too much detail. The key point here is now that we have a whole range of separation techniques in front of our MS. A lot of these are chromatography based. Some of them are denaturing, like classical reverse phase, where you have organic solvents and acids, but you can also separate proteins in a native folded conformation using tools shown here. These are routinely used in our lab. And of course, what I will focus on today in the last part of my talk is the capillary electrophoresis that you can do charge variant separation with. We then combine that with our mass spec platform. We use Orbitraps because they are really excellent tools for, especially for doing intact mass analysis, which is roughly 90% of all the analysis we do per year. I'll show you some details on that. But it's also important to have the right software platform to not only control acquisition, but also data storage. We use Chromeleon, so everything, all the instruments are controlled by Chromeleon, and the data is stored in Chromeleon in a cloud-based environment. That means you can access it from anywhere, basically. And you have you have searchability and traceability because we ensure that we have all the right metadata associated with our projects. I'll show you some examples of that later on. When we do more advanced data processing, we move into the protein metrics platform. Again, we're using Biosphere, so it's a cloud-based platform that's easily accessible from anywhere, and all the different users can access the system simultaneously. And also these two systems, the Chromeleon and the Biosphere, they talk together. I will show you how that works in practice. This slide shows the number of MS analysis that we have been doing per year. And in 2018, we introduced the native MS, primarily size exclusion MS, but also charge variant separation using cation exchange chromatography. And since then, the number of analyses have really gone up. Because people understood, our colleagues understood that we're now able to support products much more efficiently, and we can really, pull out the impurities that you see with your different separation techniques and then get a mass signature very efficiently. So it's become an increasingly important platform. One of the tools for that is the SIP chip, which I will be talking about soon. This is just an overview of how we've kind of made a transition from a time-of-flight-based detection to Orbitrap detection that has really been driven by, as I mentioned previously, the fact that we do 90% of what we do is intact mass analysis, and the Orbitrap platform is really powerful for that, even when working with really large molecules. Looking at the software, we've had a transition from, of course, we use different software to control the different hardwares from the different manufacturers. But the more important point here is that we've gone from these standalone PCs, acquisition PCs, controlling individual instruments, like we were using Chromeleon client, so that's a standalone PC standing next to instrument, controlling that, until 21. For the fusion, our tribrid, we had to use Excalibur. We really wanted to move that into the Chromeleon environment because it's a much more efficient environment for analyzing chromatographic traces and really comparing large number of samples, annotating your peaks. But the tribrids weren't supported until 2022, so we could finally do the migration there. So that's primarily software controlling our platforms. If we then look at software we use for data processing, data mining, and reporting, we've gone again from standalone biologics that was primarily for peptide mapping. Standalone PC, which only one user could use at a time. Actually, we had two of those, so two users, but only one user at a time. We then quickly switched to intact mass analysis from protein metrics as well. Both these tools were kind of came under the BIOS umbrella. It's still a single-use PCs, but it's kind of, you had all the different tools under one platform. And finally, in 21, we then did the transition to Biosphere. That's basically a cloud-based version where you have all the protein metrics tools stored in a cloud accessible way, just like Chromeleon. And the really nice thing is now that these systems talk together. So that means all the raw data we are compiling in Chromeleon automatically gets synchronized to Biosphere. In the old days, when we had the clients, we would have to export the data to a network drive, and then we would have to point to that network drive and then process the data that way. But you can imagine as the number of analysis goes up, you start to get pressure in terms of data processing and reporting in an efficient way. And so that's why the transition to a cloud-based solution where all the data flows seamlessly and automatically gets synchronized has really been instrumental in order for us to be able to process and store data, report data in an efficient manner. This is just another view. It's a bit very busy slide, sorry about that, but the point is everything is now stored in the cloud and accessible from anywhere. And in the labs, we have the different instrument PCs, like we have 4 PCs controlling our 4 Orbitraps. In the MS lab, they're feeding data into the Chromeleon enterprise environment. We also have our LC lab with roughly 10 LC systems hooked up there, controlled by 5 lab PCs. So each PC controls two LC instruments. Basically, all the data here is being fed into the Chromeleon. That's being synchronized to Biosphere. So there's absolutely no manual data transfer or renaming or exporting, and that's really important. And another important thing, when we access these systems, we either use the client, so you could install a Biosphere client on your local PC, but there would be some traffic, and we want to avoid that. So we basically have a remote desktop server. That means that the software application is actually running in the cloud. It's just the user interface that gets mirrored to your PC. So for instance, when I go traveling and I'm sitting in a hotel with a poor internet connection, I can still process data and report data because all the heavy lifting is done inside the cloud. This is just an example of what an intact mass sequence looks like. And I just want to point out the metadata part of it, which is really important when you want traceability and being able to retrieve old historical data. If one of our colleagues suddenly wants to know a new information of a sample we analyzed a year ago, we ensure that we have all the relevant information. For instance, we have a project column. That's the specific project that was analyzed. We have the analysis request number. That's a number that when our colleagues want us to perform an analysis, they draw this number and they associate a different analysis with it. This could, for instance, be a SEC-MS analysis. But we also have a very important functional metadata, and in this case, it's the theoretical mass of the antibody that we're analyzing. So, for instance, when we do lead selection studies, we'll analyze 400 or 4 times 96 samples typically. And then we need to see, do we observe the theoretical mass? Yes, no. And then we, because this metadata is already stored in the system with the sequence, we can very quickly get a yes/no answer in a report of whether we detect or whether the most intense mass is the expected mass or not. So it allows us to screen through these, especially the lead selection studies, in a matter of seconds to see if everything is completely off. Of course, sometimes we get the wrong theoretical masses and everything is red, and then we go back to the people providing the masses and say, sorry, we need new masses. That's the way it goes sometimes. We also have a PETmap workflow. Here you can again see we have the project and we have again the analysis request number, but here the relevant information is what protease did you use when you did the digestion? Was it reduced or non-reduced? All this information is stored inside the system. It's searchable, it's retrievable, and it's really important when you're doing a lot of analysis that you can search and retrieve all data efficiently. This is an overview of the analysis distribution the last 2 years. And you can see we have primarily SEC-MS was around 71% of our analysis in 2021. And you can see the distribution here is changing a lot from year to year. And that's important to point out that the analysis we do is very much project driven. So if we have a specific, for instance, in 21, we were working with a fusion protein which had trouble with a disulfide linkage. It became cysteinated, and the reverse phase method was super efficient at pulling out that impurity. So we did a ton of reverse phase analysis because of that one project in 2021. And we see in 2022, we've started doing a lot more SIP chip analysis. That's become our primary charge variant analysis. And it's a dynamic situation, and it really depends on the project. And of course, sometimes you implement new technology that works really well, and then you'll see changes over time and how you implement that as well. But SEC-MS is our primary workhorse because it's such a simple and robust setup. And what about the robustness? Well, for instance, sometimes you need a new instrument, or maybe the vendor does not support your model anymore. It's getting too old. So you have to transition to new hardware from time to time. And initially we were kind of concerned about the robustness of the data we were generating. But now we've compiled data over many instrument generations. Here you can see we have 4 Orbitrap generations. These data have been collected over a 3-year period, and here you can see it's one of our reference materials. We always include that when we do SEC-MS at the start and at the end of the sequence, and in between if we have a lot of samples. And you can see the robustness of this platform is really, really solid. We have a great confidence in the reproducibility, even when we're switching from one instrument generation to another. This was intact mass analysis. If we're looking at peptide mapping, so here we have each section here, each column is a quality attribute. For instance, the first one is a deamidation at glutamine 5 on the heavy chain. And here we did over 5 instrument generations. You can see the instruments here. And again, the take-home message is what we are seeing, the trends we are seeing is quite similar between the different instrument generations. And the primary degradation pathway for this product is asparagine 92 on the light chain, which undergoes a deamidation. So that's clearly the main degradation pathway for this product. Oh yes, I forgot to mention, this is a 3-year stability study, real-time stability, so the product has been stored at 5 degrees for up to 36 months. I'm just going to go quickly to this. So these are some examples of some of the data we've been generating. SEC-MS, our primary workhorse, is kind of cool because you're combining like biophysical characterization, where you can look at aggregates and fragments. Would you have that if you have those in your product? And then for the main peak, you can do the MS readout and really assign the different product or major product variants very efficiently. So, and you're using isocratic separation, so it's really easy to set up. This is an example of charge variant analysis done using chromatography. And in this case, you can see the blue trace, that's from our stability sample, and we can see a distinct rise in basic forms. Now, because we have a mass readout, we can see that the basic 1 form has lost water, 18 Dalton. The second peak has lost 2 times 18 dollars, so 36. And combining the fact that it's migrating basic and it's losing water tells us that it's a succinimide variant formed from an aspartic acid, likely an aspartic acid residue. This is an example of the SIP chip analysis, where we see a really intense splitting up in some main and some basic forms. And again, we have an MS readout, and we can see, well, there's a shift of plus 128 for basic 1 and plus 2 times 128 for basic 2. So we know immediately that these are terminal lysines that haven't been trimmed efficiently off our molecule. Finally, there's an example of a reverse phase. This is a mixture of two antibodies, and of course, it's a little difficult to see the mass, but trust me when I say these are the correct masses for antibody 1 and 2. But then you can really go in and look at this minor peak in front of antibody 2, and you will see this is antibody 2 shifted 16 Dalton. So we know this is, of course, an oxidation. So the combination of different separation techniques with an MS readout is a super powerful tool for really mapping what's going on at your molecule at the intact level or subunit, if you prefer that. Yeah, it's been compiled in this publication. I'll move quickly on that. So here are some examples of how we have used other techniques. This is protein A MS, where we've put a protein A column which has high affinity for antibodies. So that means you can analyze really dirty samples and really dilute samples. In this case, this is the upstream fermentation process. So this is done at GMP scale, 2,000 liter. And when you go from left to right, it's from day 0 to harvest day in your upstream process. And if you go from bottom and up, it's sorted by increasing mass. So the lower bars represent more immature glycoforms, and the higher ones represent more mature glycoforms. And here you can compare a scale from the GMP 2,000 liter scale to a 50 liter scale done in-house at Symphogen. This was used for tox study. This is used for clinical studies, and you can see we get a kind of similar profile. But what if you actually vary a lot of these parameters? And that's what we've done here with the DOE design, where we varied the pH, temperature, and the dissolved oxygen. And you can see we start to see some dynamics here. For instance, condition 10, the top 3 bars here represent terminal glycoforms with terminal galactose. So those 3 here, they add up to something like 20% in condition 10. And if you look at a condition 8, you can see that the value is way higher. It's closer to 40%. So, you know, by changing some process parameters, you can really impact your product quality. And we're starting to look more systematically into this to support our upstream process guys, so we can, you know, end up with a desired product quality and titer and all those things you look at in your upstream process. So here's an example of challenges you can face when you do intact mass analysis. This is an antibody which has a lot of terminal lysis. I actually already showed you the same one separated by the zip chip a few slides ago. And you can see when you try to assign these, it's complicated because you have overlapping isotopic patterns, asymmetrical peaks. But what if you then remove the terminal lysine using carboxypeptidase B, There you get a really clean spectrum, and now it's super easy to assign the main glycoforms. And of course, you can remove the glycoforms, but not the terminal lysines, and then you get this profile where you can easily assign the different charge states. And now you can actually also separate these by the zip chip, which I showed on a previous slide. So this is an example where the MS resolve the charge variance, but you can of course also do it with the zip chip, as I showed previously. And finally, you can treat with both enzymes, and then you end up getting the deglycosylated mass and also the glycated. So that's the chemical addition of sugar in your culture medium to primary amines. We usually see somewhere between 10 and 20%. So this is kind of to illustrate how different sample workflows are used to address product complexities when you want to kind of look at the intact or near intact level. Another example is when you introduce an extra sulfur atom in your disulfide linkage, then you get a trisulfide. The mass shift of that is only 32 Daltons, and here you can see a simulated spectrum and the sum spectrum in gray. And you can see when you have these minor shifts in the mass, it becomes really challenging to analyze it when you're looking at the intact level. You really need a relatively high level, again, this is simulated before you start to see kind of a valley between your spectral components. So that's challenging, and if you look at real life data here, you can see what we did at the intact level. We knew we had some trisulfides in this product, but it was very difficult to resolve. So what we did was we tried it at the intact level, and we analyzed 24 samples with different levels of trisulfide, and you can see we're really only picking up the stuff that's high level, so around 20% or higher when looking at the intact level. If you then do a subunit and cleave just below the hinge region, you get a much cleaner spectral readout. And here we can really, with high efficiency, assign the trisulfide forms, even those with 2. And then it becomes much easier to quantitate the trisulfides. I forgot to mention the trisulfides always sits between the heavy chain and light chain, or between the 2 heavy chains. So it's between different chains, and therefore it makes perfect sense to do the subunit analysis, because that's where you will find the trisulfides. So moving into the SIP chip. This is our platform. So we have a table, a mobile table that's height adjustable. It's electrical, so we can adjust the height easily. And then we can move this table around, because the SIP chip will go on any of our instruments. This is our Explorers 240, where the SIP chip is not installed, but here you can see on our Explorers 480 where the SIP chip is actually installed. We have the system with the autosampler because, we do a lot of samples, so the autosampler is absolutely essential for, for efficient analysis using this platform. And this is an example of what we see when we do release testing in the QC lab. So we don't do release testing internally at Symphogen. We're a development lab. So as soon as we go into the clinic, we outsource the production and the release testing of our product. And imaging capillary isoelectric focus is one of the charge variant analysis that we always use on our specification. So when we release a GMP batch for clinical testing, it's released with a profile that looks like this. Now, the really nice thing about the SIP chip, which we can do in-house, connected to an MS readout, is that we can get a profile that's very similar to the ICE profile, especially if you just switch it. So you have kind of the same acidic basic polarity, and you can see it's fairly easy to assign the profile we are seeing. And now, of course, with the SIP chip, we have an MS readout, so we can really assign what these different charge variants are. And I will show you some examples of that later on. But initially, I just want to talk about the 98 adducts, that those of you familiar with the SIP chip may be familiar with that. And it's something we've been working on, trying to address. So I want to make that discussion here in this forum, and hopefully inspire some people to try the same if you've seen this issue as well. We were told that this could be related to the DMSO that you're using. The mass of DMSO is 78. The mass shift we see is 98. So there's something going on there. It could be an adduct of the DMSO. But in any case, we wanted to verify whether the DMSO quality was relinked to this adduct, these adducts that we are observing. So what we did initially was we tested the same sample, but with different amounts of DMSO. And indeed, we see a correlation with the adduct intensity. And we could clearly see a drop in the signal intensity when we halved the DMSO concentration. So based on this, I asked the lab technician doing this work to buy all the different MS grade DMSO out there and test them. And so that was what we did, and these were the results we got. So here you can see the different brands we've been testing, and we were very happy to observe that one of the brands had almost no adduct, and that was the ACS reagent. you can see the catalog number. I'm probably going to get scolded by some of these vendors, but it's important to get this out there. So we've now ordered multiple batches of this ACS reagent, but unfortunately it's very difficult to get different lots or different batches. They tend to be produced in bulk, so there's a lot of waiting time. So in the meantime, I'm sharing this information with colleagues and people working with the SIP chip, and hopefully, if you're working with it and you have seen this adduct issue, please test out this Sigma ACS reagent and share your observations. So I'm really hoping this is reproducible, and that so far we see the significant drop also when we leave the DMSO to age. So yeah, please look into that and share your information. So how do we use the SIP chip in our everyday life? Well, one of the issues we faced, and I already talked about that, is that we had a disulfide linkage that was not being formed correctly. Instead, you would have free cysteine in the culture medium that would kind of attach to each cysteine, forming this missing linkage with a plus 2 times 119 Daltons, so it's around 238 Daltons. And this is an example of a charge separation on the SIP chip for that particular product. And we could see we had a classical basic form, which was due to the missing pyroglue. And so if you had an N-terminal glutamine, that will usually spontaneously be converted into pyroglue, and in that process you lose a basic group. So if that's not happening, you will have an extra basic group, and then you see that is beautifully separated from the main peak when we do the SIP chip analysis. And of course, the main peak gives us exactly what we expect. We also see some acidic peak that's classical for deamidation. It's nearly this, it's the same mass more or less. But when we go out to these more acidic peaks, we actually start to pick up our cysteinated forms. So you have a deamidated form of the main peaks. You will have the same effect for the cysteinated form, where you have a minor form because they are independent modifications. So you can really see this modification here is really nicely resolved on the SIP chip, and that allows us to support the observations we made on the reverse phase, which was our initial way of resolving these cysteinated forms. So again, you can observe a lot of synergy between the different methods, and if you observe the same phenomenon on completely orthogonal separation techniques, this really gives you great confidence in your results. So yeah, we can resolve these forms with missing disulfide linkages. And this is another example where we see a lot of charge separation. This was a product that had an extra glycosylation site. And so what was going on here? Well, we could see that this product had a lot of sialic acids, or no as you see here. We detected 1 with 1, 2, 3, 4, and 5 sialic acids. So that is clearly confirmed by the mass readout we get when we do this charge variant separation. So you can see it's a very powerful way of determining at the intact level what is actually going on with your molecules and why do you observe these charge variants. And of course, the sialic acid is traditionally something you would like to avoid in your product, especially if it's in the constant region of your antibody, because that has been associated with immunogenicity. So it's really powerful when you can very effectively pull out these glycoforms at the intact level without any sample preparation. This is another example of a project analyzed by cation exchange, and it's a mixture of 2 antibodies. And here you can see we're struggling a little to separate it by ion exchange, but we can clearly see that the antibody 1 is dropping. This is, again, a real-time stability study. We can see that the acidic form is increasing, and we know that this is a deamidation. And we see that by the cation exchange. We then did the same. We verified the masses and confirmed that this was deamidation, so nearly no mass shift, a very modest mass shift around 1 to 2 Dalton. Now, if we did it on the SIP chip, we get a reverse profile, but a similar pattern. And actually now we start to get a better separation between the different product variants. We can, for instance, see that there's actually an acidic form of antibody 2 migrating in front of antibody 1. We cannot do that because we don't have the resolution on the cation exchange. But like for the cation exchange, we see a drop in the main peak and a rise in acidic forms due to deamidation, also with the SIP chip. So these methods confirm each other. And in case of the SIP chip, we can really enjoy the higher separation power that we get for this particular product. So with that, I would like to summarize. I hope I've convinced you that we now have a broad range of separation techniques that we can combine with our MS platform that allows us to really associate mass changes with the different product variants we can pull out, and that is just a very powerful way of confirming what is happening to the molecule at the intact level. And I showed you that we can use the SIP chip to pull out glycovariants, deamidations, and other forms very efficiently. And hopefully, for those of you who have been struggling with the 98% adder, please check out this Sigma ACS DMSO and see if you can reproduce our results. I'm sure 908 devices, and we would also appreciate that information. I already said it, we ordered different lots, but there's a month waiting time for that. So in the meantime, hopefully some other people can confirm what we have seen. With that, I would like to thank everybody involved. Would be very happy to answer some questions.
Before we jump into the Q&A though, we do have a couple poll questions for the audience. So I will get our next poll question here ready. All right, excellent. We have 91% for the LC fraction collection. Now we're going to go over to our Q&A. Dr. Adi Kupani, a principal scientist here at 908 Devices, will now lead our discussion for the Q&A session.
Thanks, Jessica. And hello, everyone. My name is Adi Kulkarni, and I will be conducting the Q&A session for today's webinar. Well, first of all, thank you, Dan, for a very informative webinar. And we already have a number of questions coming in from the listeners. So let's get started. The first question is, can you elaborate on when and why you chose between the different charge variant analysis methods for your MS, such as CIEX-MS or ZipChip MS? Yeah,
I mean, it really depends on where do you observe a change in product quality. And very often we observe that when we do the ice separation. And so for those examples, we would run the SIP chip clearly because it's very good at mimicking the ice profile we get. We do native MS separation on it, and it's a generic setup, so there's no method development or anything, and it's just a very nice way of getting a quick indication of what's going on in your ice profile. Now, if we had a cation exchange method on our specifications and we saw a lot of change in that, that would be our primary way to go. If we see something changing in a reverse phase profile, that is the primary way we would go in terms of characterization. So it really depends on, especially when you're dealing with products in clinical development, where you observe your modifications.
Got it. Thank you. Thank you for that. So the next question is, thanks for presenting your great work. Have you had any challenges correlating ICIF and CZE-MS profiles due to the different separation principle? So for example, one is based on the isoelectric point, whereas the other one is based on Stokes radius versus charge.
Yeah. I mean, we haven't done a systematic comparison. So the answer is no, but I can say just by judging the profiles, we almost always get something that is very similar. And that gives us, of course, confidence. at the end of the day, if you're uncertain to whether the impurities are different, we would probably go in and do some stress studies to kind of provoke that impurity we are expecting to get pulled out. But overall, I would say we get a similar profile. So our first step at identifying something that's separated by the ice in the QC lab would be to do the zip chip in our characterization lab.
Yeah, and also, like you showed in your presentation, you can go one layer down from intact, which is subunit analysis, and maybe probe the molecule further over there, right?
Yes.
Okay, so how do you link peak identity from release testing, ICIF profile, to zip chip MS peak identity? I think that's sort of related to the previous question also, right?
Yeah, it relates to the same question. I mean, it's we know what we have in the zip chip because we have the MS readout. So the question is, how do we correlate that with what we are seeing in ice, where we don't have any assignment? And I have to repeat what I said. It's about seeing the similar profile being obtained, and from that you can infer and that they are correlated. Of course, yeah, and I said you can use stress studies to confirm some of the stuff. For instance, is it really a deamidation? Then you would stress it with high pH, for instance, and see if that impacts your profile in both the ice and the sea. And I know, I mean, you can do preparative ice. There are tools out there for that. So, I mean, that would be another way to characterize those individual peaks. That is quite time consuming, is my experience. But there are tools out there to do that. The preparative fractionation with the ice separation.
Yeah, that's a great point, Dan. I think we also have collaborated with some of the other CIF vendors for fraction collection, followed by characterization of those fractions at the intact level, and that's not as efficient as running a direct sample on your system, but that can certainly be done.
I have to say that we are kind of pressed in our everyday life to analyze all the samples. And we rely so much on simplicity and robustness of the methods we use. And so it is a high priority, and that's always a challenge because you can, of course, do a lot of manual fractionation and separation, but we really want to streamline workflows and simplify them as much as possible. We don't have like one or two projects in clinical development. We have closer to 10. So there's pressure on the characterization lab.
Yeah, I'm sure a lot of attendees can relate to that. Okay, so here's a good one. Can you comment further on how you realized the plus 98 added P was related to DMSO, which only has a mass of 78?
Yeah, I mean, we heard the rumor it's a DMSO related, and I was like, oh, is that like a sodium adduct of DMSO? That's plus 21. Then we're really close. And let's just test it and let the results speak for themselves. And we saw this correlation clearly to the DMSO level, and that told us, well, it is related to something in the DMSO, and we were just very happy to find a variant that had virtually no DMSO. I hope it's reproducible. I know I shared that information with you guys and also some of the other local friends here in Copenhagen who are using the zip chip, and I'm expecting to get a feedback from you and them soon.
Yep. Yeah, the work is underway, so you'll be hear from us soon. But I think here's a question on the zip chip. Can the zip chip be used on other platforms other than thermal? And the next question is, how does zip chip differ from traditional capillary electrophoresis? So I can take the first one, you can take the second one, if that's okay.
I think you can take them both.
All right. So can the zip chip be used on other platforms? The answer is yes. Zip chip is compatible with mass specs from thermal, SciX, and Bruker. Obviously, because of the source designs on the different mass specs and other mechanical constraints, you would need a different box right? So you need a physical different front end, but the separation technology on all of these boxes remains the same. So now, how is the zip chip different from traditional CE methods? So traditionally, CE has been performed in a capillary. Zip chip is chip-based, as you can imagine from the name. So it's a microfluidic device, and it's really fast compared to the capillary-based separation methods. And if you want more information, we can provide that to you at a later time. Right, so I think there's one question related to your CIEX-MS. On slide 10, how was native CIEX-MS performed? CIEX has a lot of salt in mobile phase, which is usually not compatible with the MS instrument.
Yes, sorry, we're moving away from the zip chip. No, when we do the cation exchange, we basically use the same solvent for loading as we use in size exclusion. That's 25 millimolar ammonium acetate, pH 5.4. The separation is not based on salt, it's based on a pH gradient. That's very important to realize when we do the cation exchange MS. So the first solvent is the same we use for size exclusion. It's ammonium acetate at pH 5.4, and the second is simply 10 millimolar ammonium hydroxide that has a high pH around 10 point something. So by mixing those two, 5.4 and 10 point something solvents, you can get a pH gradient, and all the solvents used are volatile and therefore MS compatible.
We have time for just one more question. So here it is. Do you use zip chip for other applications than intact native MS analysis?
Yeah, we've tried it also for peptide mapping, and if you want to really do extremely fast peptide mapping, you can do it on the zip chip, but you will need a fast MS, I can say, because we're talking like a 10-minute separation window. And so it's really fast for that. And you can also do denaturing separation under more acidic conditions than we do. We really like the native because it gives you a nice and clean mass spectrum, because you have fewer charge states and you have lower charge states, so they're moving up into the higher MZ range where you generally have less noise. So I mean, we're in love with native MS in all its shapes and forms, and yes, the zip chip is one of the applications for native MS.
That's great. And just to expand on that, so as long as the molecule has a charge on it, you can analyze it on the zip chip because it is based on charge. So you can analyze small molecules like amino acids, you can analyze peptides, and Dan alluded to the use of zip chip for protein characterization. You can also use it for oligonucleotides. So really a vast array of molecules.
We actually did amino acid analysis in culture medium way back when you demoed the instrument, because we purchased special media without lysine and arginine where we would spike in heavy versions of those, and we could use the zip chip to confirm that there actually was no lysine and arginine in our culture medium. So it's like a quality control of the very, very expensive media we had purchased there.
I remember that. So yeah, I was saying earlier that I apologize if we didn't get through all the questions, but we are almost at the top of the hour, and I would like to thank Dan for his great presentation and Q&A session.
Thank you for inviting me. It was a pleasure, and have a great summer, everybody.
We want to thank you for your attention and attending our webinar today. Thank you, everyone. Have a great day. Bye-bye.